All right, everybody, welcome back. I'm sorry to be doing this again. I, on Saturday, took one of those at-home COVID tests, and it was unambiguously positive. And then last night, I stopped being able to smell stuff. My symptoms began on Thursday. I'm hopeful to be back sometime later this week, but plan on a Zoom lecture Wednesday unless you hear otherwise. For office hours tomorrow, I'll need to have them via Zoom, and you can also, of course, ask questions via email. If my regular office hours from 9 to 11 a.m. don't work out for you, you can email me and try to set up an alternative time. As you know or may know, you have your first exam starting tomorrow. The material covered on the exam ends today. It will be chapters 13 through 16. The exam's multiple choice in the testing center. It's about 22 questions long. Most of the questions are worth 10 points. There's about 4 of them that are worth smaller than that, but overall it adds up to about 200 points or 20% of your overall grade. Let's see. People in the past generally take somewhere between 2 and 2.5 hours on this exam, sometimes less. And then so sort of scale that according to what you know about yourself in terms of like how fast you take exams. There's no note sheet allowed. You can bring OChem models. There is a table at the back of the exam with part of the periodic table and some NMR and IR values and some pKa values. So let's see. Other than that, the exam runs through Thursday. It closes at the end of the day Thursday. Please be advised the testing center requires there to be a $5 late fee beginning at 2 p.m. on Thursday, the day the exam closes. I wish they didn't do this, but this is one of the only ways they've found effective at controlling or trying to space out crowds. They know that statistically about half of students will gladly pay $5 for a few extra hours of preparation for the exam, whereas about half of students won't. And so in that way, they're able to control how long the lines get, especially on days that big exams close. So I'm sorry about that crude crowd control effort. It is what it is. Just be advised. If you have COVID and are not able to be on campus, please contact me and we will make alternative arrangements for you to take the exam once you're back. Any other questions you have about the exam? Okay. And then today, just if you have questions throughout the lecture, feel free to type it in the chat and I will try to address the issues. It's all multiple choice. That being said, don't expect necessarily easy. In fact, as I compare scores on my multiple choice exams to what scores on my free response exams were, they're about the same. So you're not going to be drawing mechanisms or structures, but if you've mastered the material, but the questions will require you to have mastered the material to be successful. You turning in the practice exam. So, right. Understand that the practice exam is not a mimic of the exam in any way. It's much longer actually than the actual exam. It's meant to give you exam level practice on pretty much everything that we've talked about. You'll report your completion of the practice exam on Learning Suite and just check the deadline on Learning Suite for when that needs to be. I'm not sure exactly what it is right now, so I'd need to look it up later, but check that deadline and make sure if you've completed the practice exam that you indicate that you've done it. Are the questions one bubble per question, or is it multiple bubbles marked per question depending on the choices? I believe it's one answer per question. Yeah, multiple answers per question is always fun, but it's kind of a pain to set up and definitely a pain for you guys to take. So, is there a TA review? Yeah, check your email. I think one of the TAs announced a review to be held, I think today. I'm not sure on the details on that. So it should have gone out in a email to all of the class from Learning Suite. Oh, have I not opened the assignment on Learning Suite yet for the practice exam? Yeah, I'll make sure that gets open today. Sorry about that. Yeah, and Emily confirms that she'll be holding a review today at 5 p.m. Location is to be announced. Oh, and there's a Zoom link for the okay. So I see. Okay. All right. With that, we'll go ahead and get started. We're gonna spend most of our time today talking about applications of what we've been learning in biology. And I want to make sure that you are not going to get lost in the details. Every time I do this, people get a little bit worried because they're afraid, oh, I need to memorize everything. Not asking you to do that, but I am asking you to understand the chemistry. And I want to show you how so many things in biology follow the pattern that we've been looking at so far, where you have some nucleophile attack a carbonyl carbon to get a tetrahedral intermediate, and then what happens next depends on who's the good leaving group, but if things work out, we can get a substitution product. This shouldn't be a surprise. We've been talking about this since the beginning of the semester, but I'm going to refer to these types of reactions as nucleophilic acyl substitution. An acyl group is this part of a molecule, so it's a substitution reaction because we're swapping out this part of the molecule. Okay, so our first example of nucleophilic acyl substitution in biology is protein synthesis, cellular protein synthesis in the ribosome. So here's a ribbon diagram or a space-filling diagram of the three-dimensional crystal structure of the ribosome. This didn't used to be known at high resolution. It now is because of advances in structural biology. This structure, I think, came out in the early 2000s and was a major achievement. Here is the 50S subunit. Here is the 30S subunit, and they come together sort of like a sandwich. In the middle, threaded through this, would be the mRNA transcript. And then here are the familiar sites. Here is the A site, the T site, and the P site. The A site in this structure is occupied by what's called an elongation factor, which actually sits in the A site and I think pushes everything downstream. I think this is where RNA exits. The growing protein chain exits somewhere around here attached to that pRNA. So let me show you sort of what's going on inside the enzyme. First, let's talk about the pRNA. Before we do that, I need to show you what the basic sort of backbone structure of a protein is. So a protein is a polymer of amino acids, which each have a particular side chain. We are not going to worry about the identity of those side chains today. Okay, and notice that, and let me sort of label for you the three each amino acid subunit in this peptide chain. Okay, so here is an amide bond, and here is an amide bond between amino acids. And notice that you've got the amide nitrogen, the alpha carbon, the carbonyl carbon. Amide nitrogen, alpha carbon, carbonyl carbon. Amide nitrogen, alpha carbon, carbonyl carbon. So the backbone of a peptide has directionality. It has a specific order. And so we say that either end of the peptide is different. This we're going to call the N-terminus because it has a nitrogen, or the amino terminus. And then this end we're going to call the C-terminus because that's the end that has the carboxylic acid group. Now, let's just suppose that this peptide, and for our purposes, we'll just stick with this short tripeptide, understand that these chains can get hundreds of amino acids long. Let's have this blue C-terminal amino acid attached to the tRNA at the P site. And the tRNA, this is one of the phosphate groups of the tRNA, and then I'm going to draw the tRNA in a little bit of a dumb way, just because it sort of has that same shape. Understand that this is a polymer composed of nucleotides. At this end, you have the 3 bases that form the anticodon. This recognizes a specific spot in the mRNA transcript. This phosphate group is, and I don't remember whether it's 3' or 5', so I'm sorry about that, but it's one of the phosphate groups from the nucleic acid backbone, but it's tied up with the carboxylic acid in an interesting functional group that you maybe haven't seen before. We call this an acyl phosphate, and it's similar in reactivity to an anhydride. Anhydrides we've learned a little bit about, but the basic idea is you have a carbonyl attached to something that is a good leaving group. And for this acyl phosphate, all of this, the tRNA, becomes the good leaving group. Okay, so the tRNA has 2 functions, right? It's got to get the right amino acid to the right place on the mRNA transcript, and then it's got to be a leaving group. So this is the P site tRNA. This is the tRNA that carries the growing or elongating peptide chain that's going to come out of the ribosome somewhere here. So we're now going to talk about how the next amino acid comes in and does the chemistry. So for that, I'm going to use a purple color to represent the T site RNA. And I'm sorry if I'm getting A versus T site mixed up. My biology is a little bit weak, but we're focusing on the chemistry. All right. So this is the T site RNA. Again, you have an anticodon, and that anticodon is recognizing a specific spot on the mRNA transcript. And so the role of this purple tRNA is to bring an amino acid, to bring the next amino acid close to the pRNA. And I am sorry if I'm getting the various sites mixed up. The chemistry here is what matters, not necessarily which site is which. I'm sorry, I don't know that better. But anyway, So the role of the purple RNA is to bring the next amino acid, which I guess we'll label in orange maybe, to bring the next amino acid as instructed by the mRNA into position to react with this carbonyl carbon. Okay, so here is your carbonyl carbon. Here's a good leaving group. Here is going to be your nucleophile. Now I've drawn that amino group in its ammonium form, which is how it exists at physiological pH. But probably before it attacks, it needs to be in its neutral amine form. We've been uncomfortable in the past with neutral nucleophiles attacking carbonyl carbons, and that's okay. Possibly this carbonyl oxygen is being hydrogen bound, and so it's activated for attack. So we're going to have this nitrogen attack the carbonyl carbon to give us a tetrahedral intermediate. Now, stay tuned, because what we're going to do is now we're going to draw what that tetrahedral intermediate looks like. So here's the green tRNA. Here's the purple tRNA. And then here's the rest of the peptide. And just to keep everything straight, I'm going to continue to label the individual subunits of this peptide as shown here. Here is the next one that we're bringing in. So there's your tetrahedral intermediate. We get that from having the amino group on this tRNA attack the carbonyl carbon. Likely at this stage there's a proton transfer reaction to remove one of the protons on the positively charged nitrogen. I don't want to draw everything again, so I'm just going to skip ahead to the step where that nitrogen is now neutral. And then recall that all of this is going to be a good leaving group. And in particular, it's definitely going to be a better good leaving group. It's definitely going to be a better leaving group than this nitrogen would be. If these electrons on the oxygen kicked down and kicked off that nitrogen, it would have to leave with a negative charge, which is way less stable than the phosphate oxygen leaving with a negative charge. So we kick off the leaving group, and then what do we have? We have a new amide bond. And again, I'm going to label each of these amino acid subunits so that we can tell who's who. Okay. So the overall effect then, as we kick off this good leaving group, is that we have made a new bond between the orange amino acid and the blue amino acid. And now my peptide chain is on the purple one instead of the green one. And at this stage, there's going to be translocation such that the purple tRNA moves down one, and that makes space for a new tRNA to come in with the next amino acid. And again, the details there I'm a little bit sketchy on, but the chemistry is just an example of nucleophile attacks carbonyl compound. You get a tetrahedral intermediate. We did proton transfer to prevent this from being a good leaving group, and then we kicked off the good leaving group having made a new amide bond. And this happens really, really, really fast in cells. This is way faster than it takes for us to even talk about it. Okay, so let me pause there for questions. Okay. I'm still watching the chat, so if anything comes up or anything occurs to you, let me know and we can go back. The next thing I want to talk about is how beta-lactam antibiotics work. Because some of you aspire, I think, in your futures to be prescribing some of these things to people. And so it's a fun story to show you some of the chemistry behind it. All right, so this is the structure of amoxicillin. You don't have to memorize that. Amoxicillin is a beta-lactam. Well, let's break that. What does that mean? Let's break that name up into a couple of parts. What does lactam mean? A regular ester is just called an ester. An ester that is cyclic is called a lactone. A regular amide is just called an amide. A cyclic amide is called a lactam. The reason this is called, so what is the cyclic amide? It's this group right here. The reason it's called a beta-lactam is because relative to the carbonyl carbon, the nitrogen that's involved in the amide is on the beta carbon. So it's a beta-lactam, or a four-membered ring. And this four-membered ring is highly strained, which contributes to making the beta-lactam a better electrophile than expected. What do I mean by that? Why would this be a better electrophile than expected? Let me pause because I'm just seeing a question in the chat from James. Yeah, understanding that this proceeds via nucleophilic acyl substitution mechanism, nucleophile attacks carbonyl carbon, you get a tetrahedral intermediate, proton transfer to determine who's the good leaving group, and then the leaving group leaves as electrons kick down to make your new amide functional group. That's the key part. All right, why is the beta-lactam a better electrophile than expected? Let's go back to esters versus amides. And this is something that I think you've read in your text. It's fairly simple to understand, so I think we're going to move on. I think we're going to sort of move on pretty quickly, but let's compare esters versus amides versus, say, acid chlorides. All of these have resonance structures in which you could put a negative charge on the carbonyl carbon, a positive, or on the carbonyl oxygen, a positive charge on the carbonyl carbon. And we've used this resonance structure as one of our explanations for why carbonyls are electrophilic. And we can draw one of these resonance structures for all of our carboxylic acid derivatives. However, there's a second resonance structure in which we can use lone pair electrons on the oxygen or the nitrogen or the chlorine. Oops. To donate electron density into the carbonyl carbon so that there's no longer a positive charge there. Okay, and you could imagine that donating electron density into the carbonyl carbon so that there's no longer a positive charge there would tend to make the carbonyl carbon less electron poor and therefore less electrophilic. So this resonance structure is possible for both esters and acid chlorides and as well as amides, but the resonance structure is better for the amide than it is for the ester than it is for the acid chloride. And the reason for that is nitrogen is a better electron donor than oxygen because nitrogen's less electronegative than oxygen is. This resonance structure matters more for the amide than for the ester, and therefore amides are less electrophilic. So you would not normally expect an amide bond to be a very great electrophile, and this is part of the reason why our proteins are stable. However, the high strain of this beta-lactam is such that the relief of that strain pays for how difficult it is to attack that carbonyl carbon. Does it always have to be another cyclic structure on that beta carbon, or can it be something else? In most beta-lactam antibiotics, there is another ring here. Sometimes it's 6-membered, sometimes it's 5-membered. And that fused ring system probably also contributes to the ring strain. Okay, so we're going to talk about how these antibiotics work, but it's important for you to realize that beta-lactam antibiotics are reactive at the carbonyl carbon. That's going to be important later. Okay, so we've talked about structure. Now we're going to talk about the process that beta-lactam antibiotics inhibit. In doing so, I'm going to show you a lot of large structures, and you're going to be tempted to be scared and overwhelmed. The structures are so large that I can't just draw them, and that's why I've written them down before. I'm going to take notes over these structures, and those notes will be available on Learning Suite. Focus on the key chemical steps, and feel free to abbreviate whatever else you need to. Gram-positive bacteria have a peptidoglycan cell wall, and beta-lactam antibiotics inhibit bacterial cell wall synthesis. So what I'm showing you here is a couple of the subunits of the bacterial cell wall, which is made up of peptidoglycan molecules. A peptidoglycan is a molecule that has a carbohydrate portion, that's the glycan part, and then a peptide portion, the peptido part. So here is the carbohydrate portion. These are 6-membered rings. You can see the hemiacetal or the acetal carbon there and there. Oxygen's part of the ring. One of the oxygens of this carbohydrate, and I'm sorry, the squiggly lines indicate that there are other carbohydrates here and here, in sort of this repeating unit of 2 carbohydrates where one of them has this long chain. On one of the oxygens of the second sugar in this carbohydrate chain, we have a peptide chain. All right, so that's here. Okay, And actually, we'll just focus in on this part, okay? All right, so you have a branching peptide, a peptide that branches off the glycan portion. And then one of the side chains in this peptide, blue peptide chain, is itself linked to another chain of peptides, another chain of amino acids, which I'm going to highlight here. Each of these, if you notice, there's these are amino acids. Here's the nitrogen, alpha carbon, carbonyl carbon, and so on. Oops. But you'll notice there are no side chains here. The amino acid that lacks a side chain is called glycine. There are 5 of these. So this is called a pentaglycine crosslink. And the reason it's called a crosslink is because it connects the blue peptide chain from one peptidoglycan molecule to the blue peptide chain of another peptidoglycan molecule. So here's the carbohydrate portion again, here's the peptide portion. And then that blue peptide on the second peptidoglycan molecule also has a pentaglycine crosslinker with a nitrogen that's available to be used as a nucleophile in making another crosslink. So what we want to focus on today is, where does this bond come from, the crosslink between the blue peptide chain on one peptidoglycan and the pentaglycine crosslinker on the other? And then how do beta-lactam antibiotics interfere with that? Okay? All right, so what we're going to show you now is a peptidoglycan molecule before the crosslink is made. All right. And there's a little bit of nomenclature here. If you care about sugar names or amino acid names, the names are there. It's not important for our discussion. Here is the blue peptide chain, and here is the pentaglycine crosslinker, which branches off the blue peptide chain. One interesting feature to be aware of in the blue peptide chain is the stereochemistry of some of the amino acids. When we talked about proteins, the amino acids that nature uses all have this stereochemistry, and that stereochemistry is called L. For most of the amino acids, that's S, absolute stereochemistry. The term L refers back to as an archaic way of talking about stereochemistry that we don't need to worry about. But the amino acids in your proteins are all L. Much less common to encounter D amino acids, which are the enantiomers. They have the opposite stereochemistry. But in this peptidoglycan chain, you've got a couple of key D amino acids. This D-alanine and that D-alanine. Those are amino acids. They're called alanine because they have methyl groups as side chains, but their stereochemistry is D. And that's important for a reason that we'll talk about later. But if you go back and look at what things are going to look like after we've made the crosslink, look at this portion of the molecule where we have the pentaglycine red chain bonded through an amide bonds to the blue chain of a peptidoglycan. There's only one D-alanine. The other is missing, which means it must have been displaced as a leaving group. So the enzyme that catalyzes bacterial cell wall synthesis specifically recognizes this D-alanine dipeptide segment of the blue peptide. Structural recognition of those two D-alanine residues, it fits in the active site. Okay? So probably we're now going to show how the enzyme catalyzes the reaction of this carbonyl carbon between the two D-alanine residues with a nitrogen nucleophile from another peptidoglycan. Okay, so probably a base from the enzyme removes a proton from the alcohol nucleophile in the enzyme active site. We saw something similar to this last time when we talked about lipase chemistry. I'm just going to go ahead and suppose that we've already done that. Nucleophile attacks carbonyl carbon to get a tetrahedral intermediate. Okay? And again, why would it attack this carbonyl carbon instead of the many others in this molecule? Because the enzyme active site recognizes this segment. Likely there's something hydrogen bonding to this carbonyl oxygen to make it more reactive. And the enzyme nucleophile is positioned such that it's only reactive towards this carbonyl carbon. So here's the tetrahedral intermediate that we form as a result. And again, I'm going to try to use our same color scheme just to help you recognize the different portions of the molecule. Here's the key tetrahedral intermediate. Likely at this stage, the nitrogen gets protonated to turn it into a good leaving group. So I'm going to just pretend that that's already happened. And with that, the oxygen can kick down and kick off D-alanine as a leaving group. And then we're going to get our peptidoglycan chain that's linked to the enzyme. Okay? Because the enzyme oxygen has replaced one of the two D-alanine residues. We saw something like this in lipases when hydrolyzing lipids, how there was an enzyme-bound ester intermediate. And we've got the same thing going on here, enzyme-bound ester intermediate. And here's the glycan portion, or the carbohydrate portion. Okay, so, and the enzyme is going to, notice we've gotten rid of the first D-alanine, and we've replaced what used to be an amide bond with an ester bond, and esters are more reactive than amides for reasons that we've talked about. So that now the nitrogen from another adjacent peptidoglycan molecule can act as a nucleophile to attack the pink carbonyl carbon in that ester-bound, enzyme-bound ester intermediate. So how would we show that? Well, we'd have the amino group lone pairs on the amine attack the carbonyl carbon to get you a tetrahedral intermediate. We've been uncomfortable in the past with a neutral nucleophile attacking a non-protonated carbonyl. Likely there's some hydrogen bonding going on in the enzyme to make this carbonyl more reactive, so we're not going to worry about that particular issue here. Once that attack happens, we will have a new tetrahedral intermediate. With the two peptidoglycan chains, with the two carbohydrate portions, the blue peptide portion on one, the pentaglycine linker on the other, and the peptide chain on the other. Okay, so here's our tetrahedral intermediate. We have the neutral nitrogen attack, so it would be positively charged. At this stage, looking at that tetrahedral intermediate, our question is, who's the good leaving group? Right now, the neutral NH2, RNH2 group would leave preferentially to the negatively charged oxygen, so probably we have proton transfer with some basic group from the enzyme to remove this acidic proton. We're just going to abbreviate that and gloss over it. Probably at this stage, either this oxygen gets protonated first, or lone pair electrons on the carbonyl oxygen kick down to negatively charged carbonyl oxygen electrons kick down to kick off the leaving group, and probably it picks up a proton on the way out to regenerate the enzyme. And in the end, you have made this, oops. And in the end, you've now cross-linked to, you've made a covalent linkage between two peptidoglycan molecules via this pentaglycine cross-linker. And this is the new bond that we made. All right, Noah, you say, why wouldn't the negatively charged oxygen on the end of the peptide chain get protonated and act as the leaving group? I guess, do you mean way back here? I'm not sure I understand. No, if you can tell me for the first part of the reaction right here. You mean this negatively charged oxygen? Sure, that could get protonated, but this portion of the molecule, that oxygen, is not involved in the tetrahedral intermediate. So the only leaving group you have is either the enzyme oxygen or this nitrogen, and if we protonate that nitrogen first, it will be a better leaving group. Oh, I see what you're saying. Why don't we do what we did with imine chemistry, where maybe we protonate this oxygen and then these electrons kick down and kick that group off? Yeah, that is an interesting possibility, but it turns out the resulting amide that we make from doing the reaction in this direction is more stable than the functional group we would get if we do what you're describing. Sorry about that. Sorry I was confused. Okay, so this is our peptidoglycan that's now cross-linked. So this is what bacteria are doing all the time to reinforce their cell walls. Now let's talk about what the beta-lactam antibiotics do. The idea is that the beta-lactam antibiotic fits into the enzyme active site in the same, you remember we said this D-alanine dipeptide is a structural recognition motif, meaning the enzyme fits tightly around that D-alanine dipeptide. The beta-lactam antibiotic fits in the enzyme active site like the two D-alanines do. And that resemblance, that structural resemblance, is not necessarily clear. Just looking at the molecules like this, in order to see it, you need to look at a three-dimensional structure of the enzyme bound to the D-alanine dipeptide versus the amoxicillin. But if you accept the amoxicillin can fit in the enzyme active site instead of the D-alanine, If it fits there, it can prevent this portion from getting in the enzyme and being activated to be attacked and form a crosslink. But the other thing that happens is that once amoxicillin is in the enzyme active site, and let's just imagine that a base removes a proton from the enzyme oxygen. Now that negatively charged oxygen can attack the carbonyl carbon of the beta-lactam, which I've just told you is more reactive than your typical run-of-the-mill amide. That would generate a tetrahedral intermediate that would look like this. And so as electrons kick down, we can actually break the amide bond. Likely, instead of having a negatively charged nitrogen leave, that nitrogen picks up a proton on the way out, or it could be protonated first. But either way, it's very thermodynamically favorable for the beta-lactam ring to open up. I'm sorry, I'm getting a bit ahead of myself. So this is the ring we used to have. Now, the key OH group in the enzyme is tied up in an ester with what used to be my beta-lactam. And so my enzyme is now inactivated because, remember, it's that enzyme OH group that gets the process started whereby the negatively charged oxygen attacks the reactive dealanine carbonyl carbon to get a tetrahedral intermediate, and we kick things off, and then another and then the pentaglycine nucleophile from another peptidoglycan can come in. By modifying the OH group in the enzyme with the beta-lactam antibiotic, we neutralize the power of that enzyme to catalyze the reaction. In some ways, you could call this amoxicillin a suicide inhibitor because it acylates the key OH group in the enzyme and stops the enzyme from working. Okay, questions about that general picture so far? Students in the first hour asked a couple of questions about resistance to beta-lactam antibiotics. There are multiple mechanisms of resistance. One is simply to have an enzyme that will hydrolyze the beta-lactam before it gets to the enzyme active site. This resistance gene is called a beta-lactamase, and essentially it hydrolyzes this beta-lactam amide bond before the antibiotic gets to the bacterial cell wall synthesis enzyme active site. So if you can hydrolyze it prematurely, that's one way. Another way is efflux, meaning bacteria have a way of taking drugs that are diffused inside them and then pumping them out so that they don't hang around longer. And there are other resistance mechanisms available too. But that's hydrolyzing the beta-lactam and then pumping it out are two of the most common. What else? So the reason that these beta-lactam antibiotics kill the bacteria is they prevent cross-linking of the peptidoglycan, and that leads the bacterial cell wall to be weaker, leakier, and more readily cleared by your immune system. Other questions? Okay, well, with that, that concludes everything for the exam that begins tomorrow. I will probably have to have office hours via Zoom tomorrow, 9 to 11. And if any of you have questions that you want to ask after class, I can hang around here for a few minutes. So with that, good luck and have a great day.